Thanks for joining, everyone. Today's podcast is with Dr. Marcus Peter, a biologist and researcher at the Northwestern Feinberg School of Medicine. Dr. Peter and I will be discussing his innovative work studying toxic RNAs and RNA interference to develop novel forms of cancer treatments. Marcus, thanks so much for joining me today on this podcast where we're going to be talking about your work in cancer. You have some very novel ideas about ways that we can attack cancer and ways that we may be able to cure cancer. Can you tell me a little bit about your work and how all this started?
Jesse, thanks for having me on. So we study cancer, and I'm a biologist by training, and cancer has been looked at and first discovered, of course, by physicians. And a doctor, of course, looks at cancer as in any other disease, looks at the patient, what's wrong with you, and he compares the patient to normal people that don't have symptoms. And then the doctor tries to identify what's wrong, as with any disease, and then tries to fix you, tries to come up with some medicine, some remedy that will make you better. The problem that I came to the conclusion a few years back is that works for many diseases, but not so much for cancer.
What is really different about cancer that differentiates from some of the other conditions?
There's been this, how many diseases is cancer? And it depends on how you look at it. You could either say it's multiple diseases, breast cancer, colon cancer, but you can also look at it from a point of what do they all share, these aberrant, these sick cells? And then it becomes to be one disease. And then it's a disease of the genes. Every cancer shares one property, and that's the only thing that all cancers share. They are sick inside their genome. They acquire mutations, and that causes change in the cells, which make them aggressive and metastasize, and they can eventually kill you.
And that's a commonality of all cancer, sort of, I guess, the fundamental mechanism of cancer is a genetic mutation. How can we leverage that observation to think differently about cancer?
Well, the common view at the moment is once we identify all these mutations that make cancer cells different from normal cells, we can somehow attack them. And we call this targeted therapy, right? So there are certain mutations that drive cancers to be so aggressive and so abnormal. However, the issue is that even within the same tumor, one tumor of the same patient, the number of different mutations and the combination of different mutations in the same tumor can be quite high. So that the problem with targeted therapy is whenever you target one particular mutation that you identified, then it will attack those cancer cells, and they do die, but there are all these other cells that don't have those mutations, and eventually they grow out and can still kill the patient. That's why so often targeted therapy is delaying the inevitable, extends lifespan, but it doesn't really eradicate the cancer. So I think what I came to conclude in recent years is targeted therapy is great, and it has certainly a benefit, but it's probably not the magic medicine that's going to cure cancer. So then I had to ask myself, okay, can we have a different view? Can we take a different look at what cancer means? And I started as a biologist and not a physician looking at evolutionary terms. How comes that we're all here, and most of us, hopefully, cancer-free at the moment? How did we get there? So nature must have developed mechanisms that prevented cancer formation at the moment it starts. And I think all scientists agree that cancer usually arises from a single cell. So we have trillions of cells in the body, and one cell goes rogue and can eventually, in the worst case, kill us. So what are the mechanisms that nature developed to recognize the only thing that cancer really differ in from other cells, and those are those mutations I mentioned, but in general terms, is there a mechanism that allows nature to detect any cell that has large numbers of mutations, and then what would be the best course of action to make that cell die that moment? And so completely and in a way that it cannot become resistant. And I think that's what nature does all the time, and that's the mechanism we are chasing. So it's not trying to target the symptoms, to treat the symptoms, the mutations when the patients present to the clinic, and they are already sick. At that point, it gets very difficult because now you have billions of cells sometimes with hundreds and thousands of different mutations that are very difficult to tackle. And that's what makes cancer so different from many, many other diseases. The mechanism we are chasing right now, we believe we have a first glimpse of how it works, will be so fundamental because nature developed it, we hope, that it will kill all these cells that have those mutations and they cannot resist.
I guess in biochemical terms, or explain for an audience that may not do biochemistry on a day-to-day basis, what do you see as sort of the basic mechanism that the body prevents cancer? What does that actually look like? And then secondarily, how do we leverage that to either prevent or treat cancer?
Yes, I think most people will know also back from high school that the way we look, the way we move is all based on our genes, right? And the classical model is that a gene basically gives rise to production of proteins, and proteins are then the workhorses that give the cell a structure and function. And in between the proteins and the genes are the so-called RNA. And there are two major types. They basically mediate the production of proteins. And so they are coded in our genome in all these letters that people, I'm sure, are familiar with. And there are two RNAs. One is the messenger RNA, one is the tRNA that allow the cell to build the proteins. Mm-hmm. Now, in recent years, it's become clear that a very small part of the genome actually codes for proteins. But there's an enormous amount of RNA being produced. And we're talking about 90% of the genome is not producing protein, but it's become clear that most of this genome produces RNA. So if the sole function of RNA were to produce those proteins, why are 90% of them not doing it? The cell has a mechanism to use these RNAs for something else. They do something completely different. And what was discovered and resulted in a Nobel Prize, actually, is that you take some of these RNAs and you put them into cells, and they delete themselves. We call it silencing or RNA interference or RNAi. So there's a mechanism. You can take these small pieces of RNA, you can insert them into cells in different ways, and what that does is that the gene that this RNA is derived from, or the piece of RNA that is already existing in these cells, is going to get taken out. And that was a fundamental mechanism. Of course, biologists had asked themselves, why is that? And of course, as often, the prediction or conclusion was it's probably some way of defending us against viruses, because the virus is also based on DNA and some of them are RNA. But what that also gave us was a fantastic tool, because it allowed us to study the individual function of genes. We could take a piece of RNA from any gene, put it back into a cell, and delete this gene, or silence it, as we call it, and then just ask the question, how do the cells change? It's a long story, but there is a physiological version of this. It's not just we taking the RNA. It turns out that all our cells produce these RNAs. They're very small, that's why we call them microRNAs, and they have lots of different functions, and they're regulating genes negatively, just like when we do it when we put these pieces of RNAs into cells. And here's the thing. There are a number of genes in all our cells that are critical for survival of every cell. If you take a heart out of a person, we can't survive. And in a cell, it's no different. There are lots of critical components. You cannot do without them. These small microRNAs, turns out, of course, they never regulate a gene that is critical for the survival of a cell. That doesn't make sense, right? Because at the moment you take it out, the cell would die. During evolution, we must have selected against these microRNAs targeting critical survival genes. And what we found almost by accident is the dark side of the genome. It's the other side. When we actually now take certain RNAs that are not the microRNAs, but they're very similar in structure, we can deliberately target those survival genes, as it turns out. In other words, if nature developed all these small RNAs to avoid targeting survival genes because it would kill a cell, there should be a possibility for nature also to use this tool and to produce RNAs specifically that do target them. At the moment, it is advantageous for the system, for an organism, to kill cells. That's what it's based on. We can actually target these survival genes, and since this is a whole bunch of them, hundreds of them at the same time, no cell can resist. Turns out cancer cells are particularly sensitive to this mechanism. We are already using this small RNAs, introduce them using nanoparticles into mouse models of cancer, and we get growth reduction of the cancer. And the most important part is we already established, and that's also published, that the cancer cells that are treated in this way do not become resistant to this mechanism. When we take the tumors out of these mice now and study them in the tissue culture again, usually when you do that with targeted therapy and a tumor becomes resistant, it's futile to treat that tumor with the same drug again. It's now resistant. Not in our case. We take the cells out, we put the same RNAs back in, they're totally sensitive, no survivors, all cells die. So that's why we're trying to develop this into a new therapy at the moment.
So you can specifically use this through nanoparticles and target just the cancer cells in a mouse at this point, where the other cells are actually not responding to this RNA.
That's what everybody's trying to do right now. The $20,000 question is, how do you specifically target the cancer cells? Here's the thing, and I know it sounds unbelievable. We're not there yet. We're not curing cancer. But at the moment, what it appears to is that the nanoparticles we're using, they're delivering their load to every cell in the body. So we're not specifically targeting the cancer cells. It is only the cancer cells that are sensitive to this, gratifyingly. And we're trying to understand why that is. We have some ideas. We're actually thinking that the other microRNAs, the microRNAs that the cells have that avoid targeting those survival genes I mentioned, actually protecting normal cells from this effect. And that's also something that was published about 12 years ago. That's another thing that all cancer cells share. They have a global downregulation of all these microRNAs. So now this whole machinery that executes what we're doing when we put in these RNAs into the cells, it's much more sensitive. It's like a dry sponge that now sucks up these toxic RNAs, and that's why cancer cells are way more sensitive than normal cells, at least in those mouse models that we're doing right now. And of course, we're trying to develop this into a new therapy. But that's exactly what's different to most other treatments. They're trying so hard to selectively target a cancer cell. When you think about it, if cancer cells are so variable and none of them are like their neighbors, then again, that is as difficult as a targeted therapy. It's like targeted therapy all over, because no single marker has ever been discovered for any cancer that allows you to specifically target all cancer cells within a tumor in a given cancer.
Where does the state of the science currently sit on this? A novel mechanism that you're working on, something very revolutionary. Where does this sit in terms of, for me as a physician, is this something that potentially in 5 or 10 years be a new cancer therapeutic? How do you see this unfolding from where you are now into the future?
That's the most important question, and that, of course, is the question that everybody's interested in. With all the technology and sequencing the human genome and all the big promises at the moment, people want results. I understand that, and so do I. But my understanding is that there are tens of thousands of cancer researchers in this country alone, and none of them have really cured cancer yet. Unfortunately, it's the sad reality. Even though there's a lot of promising therapy and some that really cure certain types of cancer, certain fractions, take immune checkpoint therapy. That's the latest that everybody is looking into. It works miracle for certain patients, but the vast majority of them don't benefit. As a basic scientist, even though, of course, I'm asking myself that question all the time, can we actually make a difference? Will it make it into the clinic? Is it going to fail as so many other drugs before? I don't know the answer to that. All I can tell you is that, of course, since we have a number of really intriguing data, and I think an interesting concept, but in order to really develop into a new drug, I'm sure it will be about 10, 15 years. It may totally fall apart on the way, as most drugs unfortunately do, because cancer is just a very, very complicated and complex animal. But I think it's worth pursuing, because if we don't start new ideas, if we don't execute and don't test new ideas, then we're never going to be able, I think, to really make a big difference. As much as I understand the need for an immediate cure, and believe me, I get lots of emails from really desperate people, and I'm heart-wrenching every time, and it keeps me going. It drives me a lot, gives me the energy to pursue this. But of course, I cannot offer anything to them, and I'm not a physician. We're just developing this concept, but I'm in a good place here, and everybody's working towards the goal and getting this to clinical use. Yeah, absolutely.
Yeah. So what are your next steps to advance this to the next phase where it could potentially become a therapeutic in the future? Is it to do additional research about how to deliver these particles into cells, or what's next for you?
Yeah, great question. There are two aspects, of course. One is those molecules that we're using, those RNAs. Are they the end of this, or are there some that are even much more active? Obviously, every time you have something that's 10 times more active, you need 10 times less, right? And then the second part of your question is there a better way of delivery? And we're working at both of these ends. And the first part is actually very exciting. I'm totally thrilled about it, is that we had the idea that if this system really exists, if nature really developed a system that we can now utilize to attack cancer, shouldn't there be a situation where this basically is overactive? You know, we have 8 billion people on the planet. Shouldn't there be some diseases where some people have too much of it, and then maybe that results in unintended cells dying, unintended cell death, maybe some degenerative diseases? If you take it further, shouldn't those patients maybe have reduced cancer, right? So that's a paper we actually are going to submit this week. Looks really interesting. Yes, we found diseases that are caused by RNA in part, such as Huntington's, ALS, and others, some really well-known diseases, where it's been established already some years back that indeed those patients actually have a reduced cancer rate, and that is all cancers, as we would predict. Now, we identified the RNAs from these patients, and we're now using them, and they're indeed 10 to 100 times more toxic to all cancer cells. So that's the part of the drug. That was a major breakthrough for us, and it also makes conceptually sense because of the situation with these patients, right? Because on the delivery end, that is something that most pharmaceutical industry usually don't want to deliver RNAs for a number of reasons, also because they're rather large compared to the small molecules that are usual drug, like aspirin, for instance, so it's just more tedious. But if that's the way to go, I would say we all need to work together, and many people in the RNA RNA community or the microRNA community already agree on that. It's really about improving the delivery of the RNAs. But guess what? We as people, as mankind, are really good in technological development, right, aren't we? This is just a matter of time. There just needs to be more resources and more great minds that commit to delivering RNAs and modifying them in ways that become more active. Working on both these aspects, improving the drug and also improving the deliveries.
Great. Well, Marcus, thanks so much for spending the time today and talking to me and our listeners about your very important research and a lot of the breakthroughs you've already made and where you're going in the future. Where can people learn more about your work and what you're doing?
Yes, I think you can just search for my lab, Google Peter Lab, and I just checked that the number one hit that comes up is the webpage of my lab, and they can read up a little bit on the background and what else we're doing.
Great. Well, I wish you the best with this work and look forward to hopefully a podcast in the future where we can talk more about how this has evolved.
That would be great fun. Thanks for having me.